Agora nós vamos iniciar, uma palestra, a palestra da manhã. E para essa palestra nós temos o Dr. Mike Watson, um médico vacinologista. O Dr. Mike, ele é médico, com 20 anos na área de vacinas no Reino Unido. Participou do desenvolvimento de vacinas importantes como rotavírus, mRNA, varicela, zoster. Participou do desenvolvimento junto à Arcan, isso na Sanofi Pasteur, participou também do desenvolvimento na Acambis da vacina antivaríola, uma tentativa de preparo de um estoque estratégico para um eventual ataque do bioterrorismo com a vacina. Trabalhou na Organização Mundial de Saúde, Bill Melinda Gates, Gavi, entre outros organismos, e atualmente o Dr. Mike trabalha na Moderna Therapeutics, Cambridge, desenvolvendo vacinas, usando uma plataforma de vacinas sintéticas, usando o RNA mensageiro como um possível antígeno vacinal, e é isso que ele traz para a gente hoje. Quer dizer, o Dr. Mike é um excelente, tem uma história de vacinologia muito grande. Eu queria compartilhar com a gente. Por favor, Dr. Mike, seja bem-vindo.
Thank you, Marcus. Thank you. Thank you for the introduction. It's a great privilege to be here at your annual meeting, and I will do my best to do justice to that. Given that it's a technological and scientific seminar, I've divided the presentation into two bits, tasked by talking a bit about the mRNA vaccine technology, how we got to where we are, and then I'm going to move on to the preclinical and clinical data. There we go. So just starting by reminding us of the central dogma of biology. Cells have DNA, that DNA codes for mRNA, and that mRNA codes for proteins. And over the last few decades, we've managed to increasingly intervene medicinally in that dogma. It wasn't that long ago that people thought making monoclonal antibodies was tough, but actually today there are 400 approved recombinant proteins out there. Equally, it's been thought pretty challenging to produce interfering RNAs, though we now see there are 2 approved antisense RNAs out there and 40 in the pipeline. And increasingly over the last couple of years, we've seen actually people have been able to intervene at the DNA level. And with the CRISPR technology, we will expect to see an awful lot more of that over the coming years. But to date, nobody's managed to intervene at the mRNA level, at the messenger RNA level. Why is that, and why do we think we can? So the principle was established a long time ago, and this is a good example of that, back in 1990, when naked mRNA is injected directly into mouse cells. Those mouse cells can be shown to express the protein product of their mRNA. But that story is very much like DNA. DNA was shown if you inject directly naked DNA, then you could get the cell to express. The challenge was how to scale up, how to get that to work in man. And the reason that's a challenge is 3 reasons, in fact. The first is mRNA is a big complex molecule. It's a negatively charged molecule, and it's a negatively charged molecule that has to get across the cell membrane, which is also negatively charged. So we all remember our physics. That's something that's not going to happen easily. And actually, having naked mRNA floating around intravenously or in the intracellular space is not going to last for very long. The nucleases, the RNases are going to break that down very quickly. The second is, even if your RNA does manage to get into the cell, then the cell is designed to look out, as we know, for RNA through the pattern recognition receptors, whether they be the toll-like receptors 3, 7, and 8, or RIG-I. And by and large, the arrival of foreign RNA into a cell triggers a very significant innate immune response, and that shuts off expression, shuts down the cell. And clearly that's not what you want when you're trying to use mRNA to express a protein. And finally, there we go, let's go back. And finally, even if you get your mRNA into the cell, you've got to engage that mRNA effectively and efficiently with the ribosome. And the ribosome is a complex machine, itself made up of RNA, but also made up of protein. So how does one go about getting over those barriers? The first thing you can do then is look at the chemistry of the mRNA itself, and there are a number of levels at which you can intervene on the chemistry. You can intervene at the ribose level, you can intervene at the phosphate level, and you can intervene at the base level. And depending on the changes that you make, you can bring about different impact on the mRNA. You may silence its potency. Clearly, that's not something we want to do if we're trying to express a protein in the body. You can lower the innate immune response, and I stress that's the innate immune response, and that may be something you want to do completely if you're producing a protein for a protein deficiency. It's something that you may not want to do completely if you're introducing a vaccine. You may want to keep a little bit of innate immunity in there. You can make changes that make it more resistant to nuclease-mediated degradation. But as you'll see in a moment, that's something that we've used a different approach for. And the other thing that you can do is change the off-target effects. The mRNAs that you'll see and that we use, we focused on the uracil. Uracil clearly is characteristic of RNA rather than DNA. That's the characteristic that the innate immune system focuses on to tell it's the RNA. And we find that by modifying the uridine with pseudouridine or methyl pseudouridine, you can actually lower very significantly the stimulation of the innate immune system through the toll-like receptors and the RIG-I receptors. So that's the RNA, but as I said, you've actually got to get the RNA to where you want to get it to. And there are two routes of administration that we are considering, and most people are considering. One is intravenous, and the other is directly into the tissues. The challenges intravenously are to avoid the RNA being degraded by macrophages or by serum endonucleases. And the challenge at the cellular level, whether it's come via a vein or being given directly, is for your RNA to be taken up, to be transported through the cell, and depending what you're carrying in your delivery mechanism, and you could carry DNA, you could carry mRNA, you could carry small interfering RNAs. You want to make sure that it gets to its target. siRNA getting to interfering with the RNA, your mRNA getting to the ribosome, and your DNA getting into the nucleus. And I just want to focus on this for a moment because we often hear RNA and DNA vaccines talked about in the same breath. Clearly, the challenge for DNA has been very much more significant. You've got to get the DNA from the outside all the way into the nucleus. And in the 30 years or so that people have been working on DNA vaccines, that's proved very challenging. The current state of the art for DNA vaccines is to use either electroporation, which has scaling challenges and acceptability challenges, or to use little DNA-coated gold microspheres. In all cases, the DNA vaccines have tended to need very high doses, hundreds of milligram doses. But as you'll see with the RNA, we've managed to overcome that. So, how do you get it in? How do you stop your RNA being degraded? So, the RNA is made in vitro. So we make the RNA, and then you can packaging it in a number of ways. And for those who are more interested, there is a more exhaustive review, and there was a whole volume dedicated to this in gene therapy in 2017. You can pack it in a cationic lipid. Cationic because you want to overcome that negative charge repulsion between the mRNA and the cell. So you've got a cationic moiety that's going to attach to the cell. And then you form what's called a lipoplex. The challenge with some of the work with lipoplexes is that it seems to induce quite a significant innate immune response stimulation. The The other approach that people have looked at is actually polymerizing the RNA with a cationic polymer. Again, you've got the cationic moiety to overcome the repulsion between the cell and the mRNA, and you have this polyplex. And that's an area that people are continuing to work on. But the challenge with all of this is that on the one hand, you want the presentation to be enough so that it's protected and it's stable and it gets into the cell. But once it gets into the cell, it's got to be released. So it doesn't want to be so stable that it just sits in the cell like a little plastic microsphere. And the tendency with the cationic polymers is to be somewhat overstable. You can then put the two together. You can take your polyplex, you can put it inside your cationic limit and deliver a lipopolyplex. And finally, the approach that we've taken is to put together a lipid nanoparticle, and I will show you what that is. So a lipid nanoparticle basically has the following components. You've got the particle itself is made up of a combination of cholesterol, a phospholipid, a phosphocholine phospholipid, with PEG in the surface. The PEG provides stability, but it also provides contact and adhesion to the target cell. And then your mRNA is encapsulated inside your lipid nanoparticle. And with that, you get stability in vivo. You get delivery, you bind to the cell, you get micropinocytosis and uptake of the mRNA into the cell. You get a level of innate immunity which appears to be acceptable, and I'll show you some clinical data in a minute. And what you can also then do is you can play with the makeup. So the phospholipid can be adjusted, and you can have a phospholipid that has a preference for the liver. You can have a phospholipid that has a preference for muscle tissue. You can have a phospholipid that has a preference for lymphatic tissue. So you can adjust the targeting of your RNA according to the makeup of your phospholipid. So where you get to with it is that by playing with the chemistry of the mRNA, by adjusting the formulation of your lipid nanoparticle and making sure you have a process that can reliably produce that, you can then address the engineering challenges, and it is very much an engineering challenge. And I think that's when you make comparison between mRNA and even DNA vaccines and existing vaccines. Actually, with an existing vaccine, you have to work with the pathogen, you have to grow that pathogen, and often the proof of efficacy is very much empirical. Whereas if you're working at a nucleotide level, it's much more an engineering challenge. You engineer your antigen, and then you deliver it. With this package, you can adjust to the biology, and you can adjust to the metabolic and pharmacokinetic needs that you have. With that platform, you can adjust across a range of different applications. So you can have an approach for a vaccine, as I say, which may retain a little bit more innate stimulation that you would want for a secreted protein, for instance. And you can adjust for antibodies and intratumoral use. And so we currently have 4 routes of administration for the mRNA. There's IM injection. I'll talk more about that as I talk about the vaccines. There's intracardiac injection. There's currently a phase 2 study ongoing with intracardiac mRNA at the time of coronary artery bypass grafting to look at its effect on the health of the heart afterwards. Intratumoral injection, and we have a couple of ongoing studies looking at intratumoral injection and IV injection to deliver RNA either for rare diseases where they're deficient in a protein and you use the liver as a factory to produce those replacement proteins, or indeed you can use mRNA to use the liver as a factory to produce antibodies as well. And just to give an image to those roots. This is luciferase expression. If you give intravenously, as you can see in this mouse model, the liver is lighting up as the LNPs are being taken up by the liver and they're expressing the luciferase. So if you can imagine that that's someone with a rare disease, the liver is now producing the protein that they're missing. An IM injection lighting up as the antigen is being taken up in the muscle. And if you look in more detail at that, it goes into the draining lymph nodes, and a very small amount makes it to the spleen. Subcutaneously, similarly, you can also administer the mRNA into the lungs through aerosol administration, or into the eyes if you wanted to deliver something intraocularly. And so the upshot of all of that, using the different chemistry and using the different routes of administration, you can produce both secreted proteins, whether that be some protein like relaxin or that be an antibody. And we have a chikungunya antibody, which is moving towards a phase 1 study towards the end of this year. You can produce membrane-bound antigens for vaccines. So we have, I'll talk a little bit more about this in a minute, a CMV pentamer, GB-based vaccine, HMPV, PIV3 respiratory viruses, flu, and I'll talk a lot more about that in a moment. And then we have a couple of other programs that we're doing in partnership with Merck. These are by and large B-cell antigens, although we found that the pentamer also has a very strong T-cell response. But on the oncology front, as long as you put in a T-cell antigen encoding mRNA, then you'll express a T-cell epitope. We're also able to produce virus-like particles, self-assembling virus-like particles, and we have a Zika vaccine in phase 1 at the moment and a chikungunya vaccine also in phase 1 at the moment. We also have a very exciting project, which is a personalized cancer vaccine, where biopsy from someone with cancer is taken, the mutanome is sequenced, and the top 20 epitopes are selected, and a personalized vaccine is produced against those epitopes, returned to the patient, and the patient is vaccinated. And that's just started in phase 1 at the moment. And then local signaling, whether that be vascular focused or oncologically focused. Also for the rare diseases, where there's an error in inborn metabolism in the mitochondrium, you can deliver mRNA, which induces enzymatic production, protein enzyme production, and replaces those missing enzymes. The two targets there are propionyl acidemia and methylmalonic acidemia, MMA. So I'm going to just focus now on vaccines. And just to go a little bit deeper into the mechanism of action, the injection is, if I was vaccinating you with an mRNA vaccine in an LNP formulation, you wouldn't know you were receiving anything other than normal vaccine. So there's no electroporation, there's no special device, it's just a needle and syringe. By and large, as you'll see, it's 2 doses, maybe 3 doses for some indications, but by and large, it's 2 doses. The mRNA is then uptaken by the muscle cell and by specialist antigen-presenting cell, and depending clearly on which cell it takes up, it'll be processed either as a, either in exactly the same way it would be processed if it was a viral infection. So you can imagine this simulates almost exactly a viral infection, which is again one of the other advantages. Rather than introducing an antigen from the outside, you're actually going through the process that a viral infection would take you through. So you're getting membrane-bound expression, secreted expression, but also proteasomal degradation and MHC1 expression. So that allows for a good CD8 T-cell response, providing that you have good T-cell epitopes there. And then expression in antigen-presenting cells, in MHC2, and in B cells, so we'll get good B cell and CD4 responses. So I want to talk and focus on flu. Flu because I've got some clinical data to show you on flu. It's not published yet, but we thought it'd be a good moment to share this with you. But just preclinically, what did we expect to see? So this is preclinical data from biopsy mice from non-human primates and from ferrets. And what you can see here is that if you give, let's start with the number of doses. Let's start on the right with the ferrets. You give a single dose of vaccine at day 0, and you see relatively low response with that. By the time you get to day 21, we get a good response from that first dose. The second dose is given at day 21, and you get a good second response to that. If we give a third dose at that point, there's very little incremental response to that. So that's why I said that by and large, think of this as a 2-dose vaccine platform. So good immune response in ferrets. You can see these are HAI titers. So we're seeing titers up between, up at sort of 4,000 or 5,000 there. One of the other questions, of course, about vaccines is, especially protein vaccines, is how long is your immune response going to last? If you give a pure protein antigen, you'll get maybe a good response, and then it'll drop off, and you won't hear from it again. What you can see here, and this is a Baobab sea mice, and you probably can't read this. These are very old mice. These are almost 1.5 years old, rolling around in little wheelchairs. But as you can see, a very long, sustained immune response there in the mouse model. And then finally in NHPs to get a sense of how it might work in man. Of course, flu in man is very different with the pre-existing exposure. But you can see here a good immune response with 2 doses in non-human primates. So we've done 2 clinical studies with flu. The first was with an H10N8 pandemic vaccine, and the second with the H7N9 pandemic vaccine. Why did we choose those pandemic vaccines? Why did we choose flu? The first is we wanted a flu that we knew people would be naive to. And apart from a very small number of people in Asia, we haven't been exposed to either of these 2 types. So people were naive, but there's a good body of literature which tells us what a good flu vaccine looks like. And we have I think we all know we can do better with a flu vaccine, but what's the immune response to a good flu vaccine? The second is that there is an accepted correlate of protection, HAI titers of 1 in 40 or above. And that indicates that in healthy adults, if you have a titer of 1 in 40 above, approximately 50% of people will be protected. So it's not a perfect correlate, but it's a correlate that everybody pretty much agrees on, and it's the correlate that's used annually by regulators to allow the seasonal flu vaccines to be released. So in these 2 studies, this is healthy adults. They received vaccine or placebo in a 3-to-1 ratio. One study was in Germany, the other in the US. And this was a 2-dose vaccination regime 3 weeks apart. And we looked in the very first study at a broad range of doses, and we looked at 2 routes. I won't show all the data, but what I will tell you is that while intradermal administration is more immunogenic than IM administration, it causes more redness and pain at the site of injection, which is actually exactly what has been seen with subunit influenza vaccines. So we haven't pursued an ID route, we've stuck with an intramuscular route. So these are the HAI and microneutralization data for the H10 vaccine. And what you can see is the doses along the bottom here, 25, 50, and 100 compared to placebo. And these are the microneut titers compared to placebo. So we've got titers ranging from 22 up to just under 70, and microneutralization titers from 6 up to 37. If you look then at the seroprotective percentages, you can see that 100% of the individuals who received 100 micrograms of the H10 vaccine surpassed the 1 in 40 threshold of protection. And for microneutralization, that was 87%. And in fact, because H10 is so unusual, we weren't able to use the exactly matching avian flu virus for this. We had to use a quail virus, which was slightly different. So that may account for some of the gap there. What about safety? This is a big list, but basically the takeaway on the safety is that there appears to be, and this, we're 300 or so subjects into phase 1 programs, there seems to be nothing that is characteristic of an mRNA vaccine that you don't find with other vaccines. So in terms of the nature of the adverse events, in terms of the frequency and the severity as well, it's very comparable to what you would see with most adult vaccines. So there's nothing there of concern, and there's nothing there that concerned either us or the regulators or the Safety Monitoring Committee that carefully followed these studies. Because it's a new platform, the FDA have asked us to follow these subjects out for a year. So they will all be followed out for a year for safety, but also for immunogenicity. So I want to go on to H7 now. So same table, same assays, HAI and micronewts. You can see the titers here going up to between 80 and 100. And the seroprotection rates, again, very, very high, just under 100% here and 100% here. And interestingly, here, this was on a 25 microgram dose rather than the 100 microgram dose that we saw the highest efficacy for the H10. And also, even if we went up to 50, there was no apparent increase in immunogenicity. The other interesting thing was if you look at most flu vaccines, after the first dose of a pandemic vaccine, you see very little immune response. We did, in these vaccines, actually see some immune response even after the first dose. And the significance of that I'll come back to in a second in terms of maturation and of memory. So, what does that mean? How does that compare to other vaccines? So this is an NIAID study run 3 years ago, and comparing in 100 subjects per group a range of different H7N9 vaccines. And they compared subunit HA alone, and that's 45 micrograms as first and second dose. They compared it to 15 micrograms with ASO3, which is a squalene-based adjuvant, and also the other squalene-based adjuvant, MF59. And what you can see is if we look at the same titers, this is HAI titers, that's 80, that's 50, that's 9 with the HA alone. Sorry, this is percentage, excuse me, this is percentage above 40. And there's 96%. So you can say that in terms of seroprotective levels, that we're at or above the best adjuvanted vaccine. And that's not to say we're fantastic. That was a really important first step, given the history of DNA. As you know, DNA vaccines have been trying for many years, but repeatedly DNA vaccines tend to come in below existing vaccines. So it's very important to start to know that this platform can do as well as existing platforms. And you can see the same for the GMTs, 100, 29, 7, and 100. And then in micronewt, seroprotective titers, percentage, 88%, 70%, 18%, 90%, 96%. And titers here, 90, 40, 13, and 80. So very comparable to the best in there. And again, this is the H7, very similar tolerability profile, nothing in the nature, the frequency, or the severity of those adverse events that give rise for concern or have given rise for concern. So I want to touch on the question of durability of immune response. We were really concerned that if we were delivering just a protein that we wouldn't have a durable immune response. So we did two things. The first thing was that in the H10 and H7 studies, we followed the subjects out, as I said, for a year. This is data out from 6 months. And what you can see is that even out at 6 months, we're seeing GMTs that are above baseline. They're not stellar, but they're above baseline. But as you know, one of the more compelling ways of assessing priming, memory, and durability is actually to give a challenge dose later on to simulate an infection and see whether the immune response is rapid and is high. So we did that. We had a subgroup in the study where we gave a single dose at baseline, and then we waited 6 months, and then we gave them the second dose at 6 months to compare the immune response at 6 months to an immune response for a second dose given at 3 weeks. And those are these data here, and I'm going to walk you through these. So this is for H7, the 2 dose levels, 10 micrograms and 25 micrograms. And these are the HAI and the micronewt titers. And what you can see here is this is the group in all of them that had their doses at 0 and 21 days. And this is the group that had their doses at 0 and 6 months. And so there are a couple of nice things, some of them not related, but one of the nice things to see is that even in the low dose group where they had their doses at 0 and 3 weeks, we see actually the GMTs continue to rise out to 3 months. And that's very promising in terms of maturation and priming. And at the 6 months, though, we didn't see that after a single dose. We didn't see very much. But if now, where are we? Out at where's my 6 month data? Here we go. If we look at that, so this is just the single dose HAI GMTs. If we now, out at 6 months, we gave them a second dose, and what you can see here is that the GMTs 8 days after that second dose, so very soon after that second dose, are at 73 compared to 18 or after their first dose. So significantly higher. Now, there's a caveat here, is that only 3 subjects came back at 6 months. Most of them got bored and went away. Okay? So these are the subjects that came back. It's not the subjects that we chose, but this is the 3 subjects that decided they were going to stay in the study. At the 25 microgram dose, out at 6 months, you can see that the day 8 titers, just 1 week after the second dose, are very significantly higher than they were after the second dose earlier on. And the same with the micronewts, but really some very high microneutralization titers here. These are small numbers. This is a small study, but the takeaway is that with the follow-up at 6 months and with this re-challenge data, the suggestion is that this induces good priming and good immune memory, and that's important. So I want to switch tracks, and so that's flu vaccine. There are lots of flu vaccines, and yes, we'd all like a better flu vaccine, but actually what else is possible with mRNA? And one of the other things that's possible with mRNA is to express antigens in the body that have proven very hard to make outside the body, and CMV is a great example. So CMV, people are pretty clear that there are 2 important antigens for CMV. There's GB, and GB has been in a study. It was in a study a few years ago, and it showed 50% efficacy, which waned after a year. And GB is the protein involved in viral fusion to the epithelial cell. But there's another very important protein, which is the pentamer, which is involved in the initial attachment. And the belief is that that's absolutely critical, but people have found it very, very difficult to make a well-formed pentamer, because as the name suggests, it's a pentamer. It's 5 proteins put together. What we find with the mRNA is that if you deliver the mRNA, the body does exactly as it would do with a viral infection and produces a very, very nice pentamer. Clearly this is a picture, but what happens in the lab, so this is an animal study. These are animals that are vaccinated either with pentamer alone or with pentamer plus the GB that I talked about earlier at a couple of doses. And what you're seeing here is neutralization titers, so CMV neutralization titers here. And they're compared with the neutralization titers that you find in cytogam. Cytogam is hyperimmune globulin taken from pre-exposed individuals and used to treat or to protect high-risk individuals. So we're seeing very high titers of neutralizing antibodies. So I'm going to wind up there and just to give you a summary of where we are. So one of the nice things about mRNA, as I said, is you don't have to work with the pathogen. We don't have to touch the pathogen. We don't have to grow anything. We don't have to, no fermentation, no cell adaptation, none of that. All we have to do is to make sure that we have picked the right antigen, we can get the antigen in, we can get the antigen expressed, and we can stimulate immune responses that are a level that we want. And that allows us, in the discovery phase, to move very, very quickly. Most of us are used to a standard vaccine, as you've gone through all the pathogen and the growth work, to take 3-5 years and longer. By and large, we find that within about 12 months, we can generally get to clinic if we need to. And an example of that was Zika. So we had BARDA funding for Zika. We took 12 months to get to clinic, which turned out to be a bit fast. And we discovered that the leader sequence we had in there was not optimal. So we've changed it out. We've put in a new leader sequence, and there's another Zika vaccine heading back to clinic in the near future. Other than that, we have a CMV vaccine in phase 1. We have an HMPV PIV3, so respiratory pathogen vaccine, also in phase 1. A couple of targets partnered with Merck, one of which is in phase 1. The flu vaccines that I've talked about, Zika I've talked about, and a chikungunya vaccine, which is in phase 1, and we've got data coming. I know chikungunya is something of concern in this part of the world, and we'll be sharing those data as soon as we have them, in case that's something that we should be working together on. I talked briefly about some of the other work we're doing. This is the personalized cancer vaccine I talked about. The KRAS, OX40, and the triplet. Again, these are intratumoral mRNA therapies, 2 cardiovascular indications, and some rare diseases. So clearly, as for all of us, this is not the work of a few people. This is work of a lot of people, and this is some of the team in Cambridge in the US. So I'm going to stop there and thank you.
Nós temos em torno de 40 minutos para debate, então podemos fazer as perguntas e aguardar respostas e debater, ou aproveitar esse tempo máximo. Acho que o Mike trouxe uma apresentação interessante, muda o paradigma de vacinas, né? Vacinas, até hoje, o que tem no mundo são vacinas, uma linha biotecnológica, essa é uma linha mais quase que sintética, vou dizer assim, seria uma vacina sintética, então é uma coisa extremamente interessante, extremamente inovadora no campo de vacinologia. Então, eu abro para pergunta, quem teria pergunta. As perguntas podem ser em português, sempre usando o microfone, mas, se quiser falar em inglês, não tem problema algum. Obrigado.
Hi, Mike.
Hi.
Ok, Helena, por favor.
Can you hear me?
I can.
Yes. Thank you very much for your presentation. Very interesting platform, very promising. And I have some doubts in the technology. So after the injection, how long the RNA express inside the cell? And when we compare with another platform like SAM, the RNA is expressing longer, or is the same, or not? And I have a doubt about your results in influenza. Did you mention that we have a group in age 10 in the clinical trials with 400 micrograms? If I'm not wrong, in your 400
yes.
400
yes.
micrograms. But did you mention the results?
We'll talk about that, yeah, okay.
I have a doubt about that. And your best result was with 100 micrograms. Is economic viable, the process? Thank you.
Okay, so 4 questions, if I heard right. The first question, remind me, was on the... how long is it expressed for? So the expression, it depends partly on the lipid. So the lipid can have a residency time which can range from a few hours to many days. So that will partly dictate the expression time, but we tried to measure it. It looks like each RNA probably goes through about 30 to 50 rounds of replication within the ribosome. You can actually watch it on a microscope. You can watch it happening. So it's about 30 rounds. As you can see, it's producing good immune responses. The proof will be in the pudding, and we'll see as the other phase 1 studies come out whether that's enough. In terms of the self-replicating platforms, I think that's a great question. In theory, a self-replicating approach would be good because you'd use lower doses. There are a couple of challenges. One is to have a self-replicating platform with the current technology, you need your replicon. And the alpha virus replicon is about 4 kilobases. So the bigger your RNA, the more challenging it is to make, and the more it's likely to fall apart. And so we find actually if you go above 4 or 5 kilobases overall, that starts to become a challenge. So if you're starting at 4 and you're adding in 3 or 4, you've probably got a very challenging RNA. So that's one challenge, and I suspect that the formulation delivery challenges is why we haven't seen that advance, because the first self-replicating data was published a while ago now. The other challenge there is a regulatory one. I think regulators are not too uncomfortable about putting a bit of RNA in. There's RNA, millions of copies of RNA all over our bodies all the time. As soon as you're putting something in that's actively replicating, they'll ask a lot of much more challenging questions, so you'll have to get through that. The third question was on...
Was about the dose of
the 400 dose.
Yes.
Yeah, so 400 was too high with that lipid. Now we've changed that lipid actually, and we're using a different lipid, but 400 was too high, and it caused a lot of local pain. So that's why you didn't see that going any further. So I think we did 3 doses of that and decided we'd stop that. That was a big dose. It was the first phase 1 study, so we really wanted to try and explore. Yes. That was the goal, to explore the boundaries. I think if we hadn't had a no effect at one end and a stop effect at the other end, we would have been disappointed with that study. So that was the goal of that study. And the third was about cost.
Yes.
So cost is a time and scale dependent thing. So today it's expensive because we make it through contractors and they put it together. We're opening our own facility, GMP facility, in the next couple of months. And that will bring the cost down to a certain level, but still, until we get to scale, it will be expensive. At scale, though, it won't be expensive because you're just producing mRNA. So at scale, it should fall into what people would consider an affordable cost.
Thank you, Mike. Thank you, Helena. Doutora Kira, por favor.
Yes, thank you very much, Dr. Watson, for such a very nice presentation. I would say spectacular new approach yes, so wide application of this technology is impressive. Yeah. And also so broad way to use it, no? The different way to use it. It's a very, very amazing, very interesting approach. I hope we will have a real product coming from this approach. But coming with your presentation, specifically, I have some question related with you showed that you have adjuvant there for H7 flu vaccine, but you didn't mention adjuvant. No? Right. Is it possible to say which adjuvant? But for the H10, you didn't use adjuvant, or you didn't show the difference you have there. If you can elaborate a little bit on this, I would like. Also, maybe it's too naive question, but is it possible to elaborate the comparison using classical flu vaccine production technology and this new production technology relates to yield, for example. Okay, I just want to see the differences in the yield and the other issues related with production, the comparison on these different issues. It is amazing also that you showed in the clinical trial that the major reaction you had there was pain. What is there to have the pain as major event that in comparison with the controlled group, no? Another question is related with your procedure and the vaccine clinical trial for the flu. Are you thinking to have a challenge studies? Because, you know, antibody maybe is not enough. And another question is that looks like the antibody titer drops very fast. After 6 months, you have a very, I don't know whether it's very low, but it comes down very fast after 6 months. You know, it doesn't mean that we have a technology that the protection will be longer than 1 year, or because the current vaccine you must immunize each year. You don't. The expectation is to have vaccine that will protect longer than 1 year. You know what it does mean. Another issue we have there for H7 is looks like is really dependent on the concentration of antigen. The 10 microgram, you had the one response, 25 was higher. I don't know if you have a 50 or 100, which will be much better answer in this. There you didn't show that the difference. Is that possible? Maybe you can improve the answer on this. And of course, we expect to have product from this, all this. Thank you.
Thank you, Dr. Akira.
Thank you. So I counted 9 questions there. I'm going to start with the first and the fourth, the question of adjuvant. So there's no adjuvant added in there. It's been a big discussion with the regulators about should we think of this as an adjuvanted vaccine or not. And the way we're thinking about it is actually all vaccines have some intrinsic immunostimulatory characteristics. If it's a pure protein, they're not very good, but if it's a diphtheria toxoid or a whole cell pertussis, it's very good. What seems to be happening here is that the cationic lipid nanoparticle is a very nice immunostimulatory molecule. It's the right size, it's the right size of VLP, and we think it's sticking very nicely to the antigen-presenting cell. So there's an intrinsic immunostimulatory activity, which means we don't have to add anything extra in to that. And an adjuvant is clearly when your intrinsic immunostimulatory activity is not good enough and you have to add something else in. So the platform as it is seems to work very nicely without an adjuvant added in. And if you think about this biology, it's not far from, mean, it's not far from a live viral vaccine, really. It's not replicating, but it is expressing the antigen, and you've got something that's immunizing. And that's why relating that to your question on pain, I suspect that's the link. You're putting in a lipid which is immunostimulatory and induces a local reaction, and that's the pain. That's our working hypothesis at the moment. So I think that was very well spotted, but I think that's exactly right. In terms of comparison of classical versus this approach to flu, there are a number of reasons why this might offer an advantage. The first is potentially for a seasonal flu vaccine, you could decide on your strain later. So by deciding later, you're likely to be closer to the circulating strain, is the first. Second, today when you select a strain to go into eggs, you have to select an egg-adapted strain. And that's now slightly different often from the wild-type strain. So you wouldn't have to do that with mRNA. You could stick to the exact sequence of the circulating HA. The third piece then is there's a nice publication last year showing that as you grow flu in eggs, you force mutations by growing it in eggs. And you're now shifting away. So mRNA is, by deciding later, by not going to egg-adapted and by not having to grow in eggs, you're probably closer to the antigen sequence. The next question is, what else would you put in a flu vaccine? Because this could allow you to do that. And that's an important question. Would you put in a neuraminidase? Would you put in a nuclear protein? Would you put an M2 in? And I think that's something that an mRNA platform is very interesting to explore. What else you could put in order to produce a vaccine which, and that comes on to your next question, which is, well, one of your later questions, which is, could be more broadly protective. Would it be universal? I don't know. I think to be universal, one is going to have to move to something else, some other target. I think that's challenging. Challenge study, absolutely. I think for flu, that's a very good way of looking at a flu vaccine. And so I think a challenge study would be, is certainly something we'd think about. That was the antibodies, not enough. In terms of when you look at the kinetic of the immune response, it's very much the kinetic you see with current adjuvanted vaccines. As the data that I showed, though, in terms of the boostability of that after a single dose, suggests to us that it's nicely priming. And if you're getting such a good immune response at 7 days, then the chances are that it would be, even if the circulating antibodies had gone down, that it would be enough to at least affect the course of an influenza infection, if not abort it completely. But we'd have to see, actually. And finally, in terms of the higher dose, so the 50, I did show the 50 data there, I thought, for the H7. The 50 was no better than the 25. In fact, it might have been a bit worse, and there's the feeling that if you, I think, whether by too much of an inflammatory response, you actually are shutting down expression. So I think you don't, and that's the balance you have to get, I think, with the lipid and the antigen. So we don't want to shut down the immune response. I think that was most of them.
Okay, thank you, Max. I think we have time just for one question more. Two. Okay, if we are very objective, we can have to.
Okay, thank you for the talk. Very nice and interesting talk. I have a doubt. This strategy of vaccine mRNA, when inside the cells, can induce some regulatory mechanism like a microRNA that can negatively regulate this mRNA. I think that this is just a comment. mRNA strategy seems interesting to avoid some mechanism of evasion, mechanism of some virus, like flavivirus. It seems a very interesting approach.
Yeah. So I think there are a couple of ways looking to the... First of all, yes, we look very carefully at the degradation products, and what we try to make sure that the RNA is... what happens is, to just take you through the process, if someone's designing the RNA, they will first of all look up on the computer the sequences, and they will create some consensus sequences. And then those consensus sequences are put through an algorithm which further optimizes the RNA to minimize slippage, to minimize any AUGs that might cut it off somewhere else. So to ensure that the percentage of read-throughs that are complete is extremely high. So the chances of there being any sub-RNAs or sub-proteins is low and decreasing. But of course, we're looking out for that. But actually, you can turn that to your advantage. And what you can do is you can build in interfering RNAs. So if we're giving, for instance, an RNA intravenously, and you want it to be expressed in the liver rather than the spleen, or the spleen rather than the liver, or you want it to be expressed in hepatic cancer cells rather than the body of the healthy liver, you can put in small interfering RNAs that block its uptake in certain cells but not in others. So you can further target the RNA to specific cell types. So you can use the small interfering RNAs as a way of further targeting down by building that into the sequence, and that's certainly something we're doing for a couple of the indications.
Thank you, Mike. Thank you. Please, just fast, because...
Thank you also for the presentation. And I had only two questions, simple questions, considering that RNA molecule is easily degraded on cells. I want to know how long your expressed molecule is expressed in your systems. If you know about how much time is how long this molecule is induced, and in the same way, if you believe that only 2 doses of vaccine could be enough to induce the desired immune response against the major of your targets, not only influenza.
So how long, as I said earlier, it depends on the lipid, but it's probably relatively short term, a number of hours rather than days. In terms of the number of doses, great question. That was 2 doses, 3 weeks apart for flu. We're looking at 2 doses, 4 weeks apart for the sort of epidemic, Zika, chikungunya, because I think if it's, nobody needs a 3-dose, 6-month vaccine for those sort of epidemic diseases. And then for things like CMV, we'll look at more doses as well to see if we need more. So we're exploring those schedules and looking at different spacing. Yeah.
Okay. Thank you, Mike. We have to finish, but we have time. Well, Chiris, do you have a question, please? Okay.
Hello, Mike. Yes, Marcus, I have one more question very quickly. Just to touch on a different subject, you showed a little bit on your cancer vaccine, personalized. You said you identify the top 10 targets. Do you use genomics, proteomics, transcriptomics? What technology do you use to identify that first 10 targets? And what would be your model in applying that? Would you have a service where the person comes in, you get the sample, you prepare the vaccine and you inject, or would you give that to hospitals? Have you considered that?
So it's sequencing. So the cancer cells are sequenced, and the mutanome. So it's genomics to identify the targets in the mutanome. And then in terms of you're asking the right question. It's very much a logistic challenge. How do we do it?
Sure.
In the first instance, it would be send the biopsy to us, we do the sequencing, we make the vaccine, and then send it back out. But there has to be a separate, dedicated unit to doing that, and it's got to be fast. And we just get fast. In the long run, who knows? I mean, if you roll the film forward for our mRNA, you could imagine a world where we don't need to make the vaccines at all. You just have 3D RNA printers in all the surgeries and hospitals around the world, and someone just dials in their vaccine, and it just prints out the RNA and formulates it in situ. Now that's probably a bit Elon Musk and a few years down the road,
so.
but it's engineering, and I don't want to play down current vaccines, but current vaccines are really haute cuisine.
Mm-hmm.
You have a dedicated kitchen with dedicated chefs and dedicated ingredients who are able to make fantastic meals that I couldn't make in my kitchen. And they have to do it consistently, and that's an amazing achievement. But actually, RNA is much more of an engineering challenge than it is cookery. 3-star Michelin chefery versus engineering, I think.
I think the printing the RNA is probably the smallest of the problems. Then formulating it with the liposomes and all the rest could still be a challenge.
You've got to have a long-term view, haven't you?
Of course, of course. All right, thanks.
Obrigado, Sotiris. Obrigado, Mike. Obrigado pelo excelente debate. E agora eu acho que nós temos tempo durante a sessão de pôster para qualquer conversa e continuarmos. O Mike ficará aqui um tempo, o dia com a gente, então estamos abertos para